you. Good morning, everyone. Welcome to, we think it's the Goodman Cancer Research Center, but it's of course the Zoom version. So there's quite a few people that are going to be attending this workshop today. It's the second in a series of workshops that we have intended. The first one was held last year and involved mainly cancer cells and technologies to analyze cells. This time around, we're climbing the ladder and essentially going into animals, particularly mice, because these are preferred models for cancer research at this point. And we have a roster of specialized investigators who have been working with this, some of them for close to 35, 40 years. So they are the experts in many of these technologies and have basically made significant contributions to cancer research using these models. The first one of our presenters is Dr. Bill Muller. Bill is actually a former graduate of McGill University. He completed his PhD in 1986 in the Department of Microbiology and Immunology at McGill University. From there on, he went down to Harvard Medical School and studied with Phil Leader as a postdoctoral fellow. He was there 3 years, and I think this is where he developed some of the very first mouse models, transgenic mouse models in those days, to study breast cancer. He then came back to Canada and was a professor at McMaster University, and where he stayed for quite some time. I think it's something like 17 years before he decided to come and join us at McGill University with affiliations in the Department of Medicine, Biochemistry. He is a CRC chair in molecular oncology, and his laboratory is located within the Goodman Cancer Research Center. Bill has won many, many prizes over the years. Pages of his CV are actually populated by some of the prizes. And of course, there's more coming. One of the most prestigious prizes, he was a Medical Research Council of Canada, MRC scientist for many years. And he's had the chair of CRC chair in molecular oncology since his arrival at McGill. He was also named a fellow of the Royal Society of Canada in 2011. Bill is undoubtedly the expert on mouse models of breast cancer in the world. I asked him earlier if he knew how many countries had received this mice, and he's never calculated it, but for sure it's all over the place, not only in research laboratories, but in many companies. And you'll see why, because he's going to walk us through his discoveries and why these models have become the best models. So for all the speakers, we will try to devote the necessary time to your presentations, and Danielle will tell you 5 minutes before the end that there's 5 minutes remaining. For all the attendees, we ask that you put yourself on mute. If you have any questions, please place them in the chat. We will ask the questions at the end of the presentation. And Bill, Yojiro, and Mitra will remain at the end of Mitra's presentation so that other questions that may have popped up can be answered before lunch. So Bill, the floor is yours. Thank you very much for accepting all of this, and we're looking forward to your talk.
Okay, thank you very much, Nicole. Good morning, everyone. So today I'm going to talk about genetically engineered mouse models, particularly human breast cancer. And really, this is what our lab is focused on since its founding in 1989. And something I took from basically the Leader Lab when I was a postdoc, where we developed one of the first models of HER2-positive breast cancer. Now, to give you a little perspective, let me see. Breast cancer, basically it's a disease that's one of the most common cancers within human female populations. And it undergoes a whole series of interesting pathological changes, characterized initially by ductal hyperplasia, leading to what's known as ductal carcinoma in situ, which is essentially uncontrolled proliferation within a barrier of a myepithelial layer. And then eventually that's broken down where you get the pathological invasive ductal carcinoma, which can go on to metastasize to many different distal sites. So as you can see from this progression series, really modeling how this occurs in an in vivo setting is really critical to understanding the underlying pathology and molecular biology of this disease. Now, prior to the development of these models, there was extensive work on breast cancer, and at the time, they were initially characterized as hormone receptor positive or hormone receptor negative based on the estrogen receptor status. Subsequent molecular characterization allowed further subdivision into four major categories: the luminal A, the luminal B, which are both ER positive, with luminal B also on occasion being HER2 positive. Finally, in the hormone receptor negative category are the so-called HER2 enriched and the triple negative. As you can see from the lower diagram here, the clinical prognosis for the hormone receptor negative and luminal B is significantly worse than those of luminal A, which are readily treated with anti-estrogens. So there was a real pressing need to model these very aggressive forms of breast cancer. Since this initial characterization, of course, there's been further molecular diversity within the breast cancer sections, including the tumor microenvironment, such that there now could be tens of different categories of breast cancer, depending on what the epithelial and the tumor stroma reflect. Now, as you probably, this is a classic diagram which has since been added on, which basically illustrates the hallmarks of cancer. Now, as you can ascertain from this diagram, it's very difficult to actually model these hallmarks in simply a tissue culture setting, particularly when we talk about angiogenesis, tumor invasion and metastasis, and other aspects of the tumor microenvironment, including immune surveillance and metabolism. Therefore, there was a very pressing need to develop a model which could essentially mimic the in vivo characterizations of tumor progression. And of course, we turned to our favorite model, the mouse, largely because it was a mammal and it had mammary glands, but more importantly, its generational capacity was such that you could really generate generations of mice within a reasonable time frame. And the genetics of mice had obviously been worked out over the preceding decades, such that there were well-characterized strains of mice that could be subject to genetic manipulation for the generation of these models. So why make a mouse model of human cancer? Well, you really want to understand the effects of oncogene activation in the context of the whole organism. So which actually occurs in vivo in the human being. We know quite recently that the tumor itself is influenced by a host of factors that are involved in the tumor microenvironment. These include stromal fibroblasts, endothelial cells, and important immune components, which also have become recently targeted components for immunotherapy. We also can use genetics of the mouse to define the nature of cooperation between oncogene promoting oncogene drivers, as well as another class of genes known as tumor suppressors, which effectively act as the brakes on uncontrolled proliferation and gain in the in vivo setting. And finally, these preclinical models really can be used to directly address the utility of targeted therapeutics to various driving oncogenes. This component really has seen multiple applications with respect to developing novel therapeutics that target various driver oncogenes in mice. So to give you a perspective of how I got involved in the whole field, we take you back to the year 1987, where a clinician known as Dennis Slayman, in collaboration with a molecular biologist known as Axel Urick, showed that an EGF-related protein known as ERB2 or HER2 was amplified and overexpressed in about 20 to 30% of breast cancers. So what Slayman had shown is that in these patients, they had a particularly bad outcome. And he suggested that the HER2 oncogene was largely the driving oncogene within these mammary epithelial cells. Although the data at the time that he had only suggested correlation and not causation. Now, being a former microbiologist, I was very familiar with Koch's postulates, which state that you really have to prove that the villain driving the phenotype is actually driving the phenotype. So at the time in Phil Leder's lab, they were using a retroviral promoter known as the mouse mammary tumor virus promoter, which naturally infects mice and gives rise to mammary tumors due to an insertional oncogene mechanism. So it integrates next to putative oncogenes such as the Wnt1 and in two loci and is able to induce mammary carcinoma spontaneously in these mice. We took one aspect of that, namely the promoter region, which we knew had a high degree of tissue specificity to the mammary epithelium, and we coupled it to an oncogenic version of HER2 known as new NT. Now, this was the rat homologue of HER2 that the Weinberg lab had shown to be the driving oncogene in a neuroblastoma cell line. And we coupled that to the MMTP promoter and put at the end of this SV40 polyadenylation sites. This hybrid fusion gene was then injected into one cell mouse embryos. And in this particular case, we used embryos from the FVB inbred mouse lineage. This allowed us to actually do transplantation analysis, since the original transgenic was generated on an inbred background. Now, since the development of this particular mouse model shown in the lower panel, we showed quite dramatically that expression of this activated version of HER2 led to widespread mammary tumor formation. And it did imply that the initial observation of Slayman was indeed correct, that HER2 was in fact driving tumor genesis in this model. Now, if we follow up the story, the direct implication of these findings were subsequent development of antibodies known as Herceptin or trisomamab, which enabled it to block HER2 activation. Although it's become clear the primary activity of this antibody is through recruitment of the immune system, namely through a mechanism called ADCC, which allows immune cells to target HER2 expressing tumor cells and eliminate them. So this is now in clinical practice and widely used in the treatment of HER2 positive breast cancer. Although it's still not perfect in that only about 50% of patients actually respond significantly to this drug. So still an open question why the other half are refractory to this blocking antibody. Now, when I left Phil's lab, I went on to ask a very important question, which was more relevant to the human situation. And by that I mean is that when people looked at mutations of HER2 in breast cancer, they could not find them. It appeared to be simply overexpression of the wild type proto-oncogenic form of HER2 that was driving expression. So to get at this issue, we basically created a similar transgene in which we expressed the proto-oncogenic form of HER2, again under the control of the MMTV promoter. Now, as you can see from this Kaplan-Meier curve, what we found is that tumor onset in this strain of mice was significantly longer and less penetrant than what we observed with the original strongly activated oncogenic form of HER2. At this point, a graduate student in my lab, who is currently at the center, Peter Siegel, went on to actually show that in virtually all these tumors, there were activating mutations within the just outside the transmembrane domain in these tumors, which involve in-frame deletions, which led to receptor homodimerization and receptor activation. So this was actually quite curious that we saw such a strong selection for these mutations within this particular model, and suggested there was something more at play than we thought initially. So when we actually further characterized this particular mutational activation, we noticed that there was a naturally spliced isoform of HER2 that recapitulates these particular classes of juxtatransmembrane mutations. Now, this spliced isoform, like these spontaneous mutations, is capable of actually forming cysteine bridge dimers, which are constitutively active. So it acts in a similar molecular fashion, which begged the question why we were developing these mutations to begin with in the wild type HER2 model. And again, this is an example of a transgenic experiment actually elucidating something about the molecular biology of the receptor. What we had put in initially was a wild type HER2 cDNAs, and what cDNAs do not do very well is recapitulate splicing. So the requirement for this splice form was such that they tried to recreate forms that actually mimic biochemically what this splice form normally does, and hence the generation of these mutations. And I would point that we subsequently went on to show that in virtually every HER2 amplified breast cancer, there's about 10% of the message that is of this HER2 splice isoform lineage. And in fact, even though the ratio doesn't change between normal and tumor epithelium, the absolute levels of the receptor are elevated due to the process of amplification. So you have this constitutively active form just being elevated to much, much higher levels than it would normally be in a normal epithelial cell. And we postulated that was probably one of the mechanisms that were behind the selective gene amplification of this gene. We next turned to another system that was developed by Lou Chodosh and colleagues, which enabled us to actually put some temporal control on the expression of these transgenes. This is the so-called TET-inducible system, and it's based on bacterial genetics involving the TET repressor and the TET operator sequences. Now, by fusing the TET repressor to activation domain from a herpesvirus known as VP16, and putting in the appropriate expression vectors, we could generate a hybrid transactivator which would only function in the presence of doxycycline when doxycycline is present in the media or in the transgenic setting in the drinking water of mice. And if we coupled the TET operator to a target driving oncogene, we could, in principle, temporally upregulate the expression of that linked gene. Or if we wanted to, then we could actually de-induce these mice by simply withdrawing doxycycline from the water. And in fact, the key contribution to that was this MMTV reverse transactivator, which expressed this specifically in the mammary epithelium. So the way these systems work are really a binary system in which you have the MMTV RTA coupled to the mouse memory tumor promoter, and that's made it to a separate strain expressing the TET operator driver in a different transgenic mouse. And so when you combine the two together, you can then have a system by adding doxycycline to induce expression of the oncogene. Now, of course, this is just data that we generated recently. And what that data shows is that we generated forms of transgenic mice that express the splice form or the wild type HER2 under the control of the TET operator sequence. And by breeding each of these individual mice with these MNTV RTTA mice and then adding doxycycline, we could induce expression of these different isoforms. And in both cases, we found that they could drive fairly rapid tumor formation, very similar, in fact, to the original point-activated form. And when we look at the absolute levels of expression of the transgene, they're dramatically upregulated, indicating that that this TET system really does allow for very dramatic upregulation of linked transgenes. More importantly, we can use the system. They ask the question whether sustained expression of the oncogene in question is required for maintenance of the tumor. And in the lower graph, we see that we've induced animals from both strains to develop tumors to a particular tumor volume. And then we withdraw doxycycline, and we can notice that in both strains, what happens is the tumors rapidly regress. Now, this is an interesting observation that we still don't have a molecular explanation. But in the wild type HER2, what we find is that tumors do recur subsequently that are HER2 negative, whereas those in the delta 16 do not. So again, it suggests that the molecular biology of these two different forms of HER2 is fundamentally different. And it also suggests that we have model systems where we can look at mechanisms of tumor recurrence following targeted oncogene inactivation. So these are really interesting developments that we definitely want to pursue in the future with these mouse models. We could also use technologies such as RNA sequencing to gather what the overall transcriptomes of these two different HER2-driven tumor types are. And what we observed is that both wild type and splice form of HER2 had dramatically different transcriptomes. So this very subtle 16 amino acid change in primary structure of HER2 had a dramatic effect on the signal output from these receptors, and indicated that really what we were dealing with were two different HER2 receptors, which activated different signaling pathways. And quite excitingly, one of the pathways that was subsequently upregulated in one of these models was the estrogen receptor, and I'll get back to that at the end of the talk. This is important because there are very few models of estrogen receptor positive breast cancer in the murine mouse model, Compodium, and this might actually constitute one of them. Now, in addition to this transcriptome difference, we could show that the end-stage tumors from the different isoforms were dramatically different in their tumor microenvironment composition. For example, in the wild-type HER2, we had a massive influx of both CD3 positive T cells as well as macrophages, whereas in the splice isoform, they were basically devoid of these infiltrating immune cells, indicating that the immune microenvironment in both is quite dramatically different. In today's terminology, we would classify the wild type HER2 as immune hot, whereas those in the delta 16 as immune suppressed or immune cold. And we're trying to get at the molecular mechanism behind driving these different microenvironmental effects. In addition to differences in the tumor immune microenvironment, we also see very much differences in the vasculature of these tumors. Whereas the vasculature in the wild type HER2 are very leaky and dispersed, those in the delta 60 are highly organized. So the relative leakiness of the vasculature is really quite different between the two models. Now, we think this has very important implications with respect to HER2 sensitivity to Herceptin. As I alluded to at the beginning of the talk, it's not clear at this point why different patients respond differently to Herceptin. And we would like to hypothesize it's the ratio of these two isoforms that is largely responsible for this difference in response. And indeed, what we can show, or what we think, is that the immune cold splice would be relatively resistant to Herceptin due to the fact that the infiltrating immune microenvironment is not capable of actually eliminating the tumor proper, whereas those in the wild type HER2 it is. So we're currently testing whether this hypothesis is of value by using Herceptin in these particular mouse models. And again, gives you the idea that you can manipulate these mouse models to determine the relative efficacy of a particular targeted therapeutic. Now, again, this has important implications with respect to another emerging field in breast cancer research, and that's the role of immunotherapy and immunocheckpoints. As I alluded to before, Herceptin really was one of the first immunotherapy agents actually developed, in that it allowed targeting of the immune components to the HER2 cells via the Fc receptor. And so, again, we think that the different forms of these HER2 isoforms may be ultimately responsible for the different responses that we actually see with respect to HER2 mediated therapy. We also are exploring, and I won't go into it, but there is also evidence that epigenetic targeting of tumor cells can actually mobilize the immune system and thus make those cells much more susceptible to HER2 targeted therapies. Now, all of the data I've shown you thus far are in some ways, artificial systems, in that we're expressing HER2 either using the TET operator sequence or using it in conjunction with a strong viral promoter such as the MMTV promoter. Now, as you well recognize, in the normal human context, HER2 is driven by its own endogenous promoter, and therefore the mechanisms that may be in play in some of the models I described may be exaggerated or not relevant in the physiological setting. Now, really, to get at this issue, over, I guess, 2 decades ago, we generated one of the first models which took advantage of the fact that we could use homologous recombination to target different exons within the HER2 gene. And so the strategy basically involved replacement of the coding exon containing the AUG of the endogenous HER2 with an activated HER2 into the first coding exon. Because we were concerned that activation of HER2 in an embryonic setting might be embryonic lethal, we placed upstream of this activating new NT LOXP-flanked neocassette, which essentially would stop transcriptional read-through of that allele, unless you express CRE. And again, the second component of this binary system is to express MMTV CRE in the memory system, thereby allowing excision of this neomycin stop cassette and expression of activated HER2 under the control of its own endogenous promoter. So in this way, we were actually forcing the activated oncogene to be expressed in its normal physiological promoter, and ask the question what effect this has on latency. Not surprisingly, what we found was that tumorigenesis in this model system, despite using a very dramatic activating mutation in HER2, was very long latency, with an average latency of 16 months. This compares to the MMTV system, which has an average latency of only 90 days. So clearly there was difference in the latency of these two models. And what was really very interesting about this model is that eventually tumors did develop that expressed significantly elevated levels of HER2 compared to the adjacent epithelium. So we asked the question whether, like human breast cancer, does genetic application occur in this model that recapitulates what occurs in human beings. And indeed, when we did southern blot analysis, we could show that the transgene encoding the activated HER2 was selectively amplified compared to the endogenous gene, and that's shown in these southern blots. Perhaps more importantly, we also showed that genes linked to HER2, very closely linked to HER2, including GRAB7 and CAB1, were co-amplified. And indeed, with Joe Gray and colleagues, we went on to show, using CGH analysis, that the amplicon is actually identical to what we see in human breast cancer. And this is a significance because both GRAB7 and CAB1 are thought to play also ancillary roles in HER2-mediated tumorigenesis. So this gets at the concept that amplicons containing a driving oncogene can have more than one contributor. And it is actually a very unique model to begin to try to resect the contribution of these individual genes in HER2-mediated tumorigenesis. We've also used this model to actually look at the role of another common loss of heterozygosity that occurs in HER2-positive breast cancer, namely 1433 sigma, and showed directly that genetic ablation of 1433 sigma can dramatically accelerate HER2-induced tumorigenesis in this Knockin model. So again, this allows us to test the concept of the interaction of a driving oncogene with a tumor suppressor and show that those are functionally important in the process of tumorigenesis. Now, we've also looked at HER2 from a concept of understanding the downstream signaling pathways that are involved in HER2-mediated tumorigenesis. As you well know, like all receptor tyrosine kinases, when they become constitutively active, they can recruit a variety of different signaling molecules, which then in turn can have diverse effects on a whole variety of cellular aspects, such as proliferation, metabolism, survival, adhesion, and invasion. Now, when we actually did this for the CSARC tyrosine kinase, we found that using a novel model in which we drive HER2 with a Cree linked to this model, we could show that tumorigenesis was quite delayed in tumor onset. This was work conducted by a postdoc in the lab, Harvey Smith, and he subsequently went on to show that these tumors had a severe proliferative disadvantage. After a lot of work, we showed that surprisingly, the core of this proliferative defect was actually metabolic in origin, reduced to essentially an interaction between an epigenetic regulator and the different aspects of mitochondrial metabolism. So we unexpectedly found that SARC played an important role in actually regulating the metabolic state of the cell by affecting essentially mitochondrial dynamics. It again illustrates the beauty of these in vivo models to really elucidate or illustrate underlying molecular mechanisms, in particular signaling defects. So in fact, what our data showed was that SARC, through its interaction with HER2, is involved in maintaining mitochondrial OXPHOS activity. And in the absence of SARC, the mitochondria fragment, and we get a generation of AMP kinase signal, which inactivates mTORC-dependent translation, which indirectly affects an epigenetic regulator that in turn can have further impacts on the biology of the tumor. So really with these model systems, you begin to see the complexity of how different signaling pathways play into both the metabolic and epigenetic state of a tumor cell. Now, another model that we developed with the aspect of trying to identify the importance of signaling pathways was the so-called polyomavirus middle T antigen model. Now, polyomavirus, as the name denotes, was initially described by Ludwig Gross And it was noteworthy that in early studies with viruses, it was shown that this virus could induce a whole array of different tumors in mice. So the question was how this occurs. And one of the principal driving oncogenes of polyomavirus is middle T antigen. This viral encoded protein is a membrane-associated protein, and in many respects resembles the C-terminal tail of an activated RTK. It is known to recruit several defined signaling pathways, including PLC gamma, PI3K kinase, and the SHIC-A adapter. In addition, it is known also to recruit the activated SRC oncogene. So in many ways, this is like a signaling nexus that we can begin to genetically dissect the role of these individual signaling pathways in tumor genesis. So back in 1992, with this knowledge in mind, we decided to create such a model and generated an MMTV polyoma middle T mouse, which is now in widespread use. Now, the initial phenotypic characterization of this mouse revealed that it basically generated widespread mammary adenocarcinoma with high rates of metastasis. So it was one of the first models where really one could begin to understand metastatic progression. However, the real advantage of this model is it really precisely recapitulates tumorigenesis that's observed in the human breast cancer progression, going from essentially a hyperplastic phase, which is ER positive, to early carcinoma, and to finally a late carcinoma that becomes highly invasive and metastatic. So we could actually, using this model system, monitor the various different steps in tumor progression. So to give you kind of a timeline of development of these different models, we've used various different aspects of the polyomavirus model to actually look at signaling pathways using the MMTV-CRE reporter. We also made mutant versions of middle T that were decoupled from various different signaling pathways and showed that these mutant middle Ts gave rise to hyperplasias, but not full-blown tumors, again implicating these signaling pathways as critical components of the tumor progression pathway. And more recently, we generated, again, a TED operator version of middle T antigen, which allowed us to look at the temporal role of middle T in different stages of tumor progression. So, like the conventional middle T model, this binary model gives rapid rise to tumors. But one really interesting exception is the penetrance of this model is not 100% like the MMTV polyamido T. And we believe this to be due to the fact that in the context of an inducible system, middle T is really a tumor antigen. So when it's induced to be expressed, it modulates the immune system. And at least in 20% of those animals, they're able to eliminate any nascent mammary tumor that arises. So this particular inducible model has the inherent advantage of actually being a model where we can closely look at the interaction of the immune system with an induced tumor antigen. So to provide more definitive evidence of that being the case, we actually crossbred that particular animal to a conditional knockout of STAT3. One thing I didn't tell you about this polyoma middle T animal is that it has a CRE recombinase coupled to the middle T antigen, which enables simultaneously inactivation of any downstream signaling system. Now, using this system, Laura Jones, a graduate student in my lab, showed that loss of STAT3 in the epithelial system led to a severe deficit in tumor formation. However, quite interesting when we actually looked at progression temporally.
Dr. Muller, you
Yeah,
have 5 minutes. Yeah, 5 minutes left.
Okay, I'm almost there. We found that the tumors did occur, but by 4 weeks, we're beginning to regress, and by 6 weeks, we're completely eliminated. And we showed this was due to the immune system being recruited to these tumors and thereby eliminating that nascent tumor. So really, and we showed that the infiltrating tumor cells were of the anti-tumor type, namely the CD8 and M1 macrophage lineage. Again, implicating this is really an important model to look at essentially immune surveillance. Subsequently, and more recently, we went on with to develop a model which could hopefully recapitulate some of the more common forms of cancers, namely the estrogen receptor positive aspects. Now, as I mentioned, estrogen receptor is implicated in about 70% all breast cancers, and patients who become resistant to tamoxifen do so in some cases by acquiring mutations within the estrogen receptor. And these lead to subsequent recurrence, primarily in the metastatic site. So we wanted to create a mouse model where we can actually mimic this process in vivo. And again, we used the same Knockin strategy in which we could introduce a coding exon downstream containing this activating mutation in the estrogen receptor, and with LOXP sites remove the normal exon 9 and replace it with this mutant exon 9. So with this MNTV increase system, we could recapitulate essentially a mouse model which expresses this activated estrogen receptor. We've actually generated mice germline that show that expression of this thing in a developmental context causes feminization of the mice, indicating that this estrogen receptor is potently activated. We're now in the process of actually crossing into a whole series of oncogene mice to see how it affects both metastatic and hormone sensitivity to various different other driver oncogenes. So with that, I would conclude and say that we're on the verge of actually developing gems that recapitulate this very important human breast cancer subtype. And with that, I can take questions.
Thank you, Bill. Would anyone have any questions? There are none in the chat, but if you have a question that you want to voice, just unmute yourself and ask it, please. So I have one question that I've been wondering about for years. Most of the breast cancer models have all been developed in the FVB background.
Right.
Is there something particular about the FVB in terms of susceptibility genes, or I don't know, organization of chromatin or anything that?
Oh yes, I mean it's an active application of the polyamide middle T model. Back in 1992, when we first generated the mouse, I was approached by Kent Unser at the NIH, and he actually crossbred it to 20 different inbred backgrounds. And using that particular sets of crosses, he quite elegantly illustrated that you could modulate the metastatic phenotype from very severe, as is the case of FEB, to even more severe in case of certain other backgrounds, or in other backgrounds completely suppress that metastatic phenotype. And Kent went on to use standard mouse genetics to map modifiers that were responsible. It turned out to be that the mouse equivalent of those modifiers were important risk factors in human breast cancer progression. These include genes involved in macrophage recruitment, like the colony stimulating factor 1 signaling access. So background is critical in all these particular experiments, and that's why we make an effort to do all our experiments on a single background, because sometimes it's ignored, but if you start mixing backgrounds, you're going to distort phenotypes. Another thing that's very apparent was when people initially took our MNTV HER2, which was on an FEB background, and placed it on a Black 6. This was initially done by the Jolicoeur lab. And they saw a very slow tumor onset compared to the rapid tumor onset we saw in the FEB. As it turns out, that's completely genetic background oriented. So for all our knockout experiments where we're actually putting conditionals on, we make a strong effort to put it onto an FAB inbred background.
Thank you. Any other questions? Okay, then Peter, did you have a question?
Yeah, so Bill, you've shown really nice data to argue that the way that the wild type ERB2 and the splice signals is very different. And so one idea is that the splice is creating cysteine bridge dimers, right? So you're forcing signaling down a homodimer. Right. So can you comment on how you think these two receptors are different from that point of view?
Yes, I mean, in fact, that's something we're testing right now. As you know, Josef Yardin, as well as others, have made a career promoting the idea that HER2 functions primarily as a heterodimer through HER2 and through HER3 and the EGF receptor. And those heterodimers are what actually does the signaling damage. What our data argue is that there's actually two forms of HER2, a HER2 homodimer and the HER2 heterodimer. So I think that largely is responsible for the differences in the signaling aspects. I would also point out that one of the other aspects was really quite surprising is the splice takes out a TACE cleavage site in the juxtatransmembrane domain, which renders it constitutively stuck on the membrane. So not only is there dimers on there, it can't be endocytosed and downregulated. So the half-life of the receptor is actually significantly higher than what we see in the wild type version of HER2. Now, the prediction that I just alluded to would be when we cross to a conditional knockout of HER3, we'll see a major impact on tumor genesis in the wild type receptor, whereas the splice will be refractive to HER3 deletion. And we're currently testing that as I speak.
From a technological point of view, I mean, you were there in the very beginning of those transgenic mice, and you've progressed, and your models have progressed with the new developments. What would you say what has been the greatest technological advance relative to giving you the best insight into the mouse models versus the human?
Yeah, I mean, the Cree-Lox recombination system comes to mind as really one of the pivotal developments that really has allowed conditional knockouts, allowing you to specifically look at tissue-specific effects of deletion of a pathway. I think this enables us to dissect stromal versus epithelial contributions of various different signaling pathways. Of course, as you're going to hear in future talks from Yujiro and Mitra, the CRISPR-Cas system is another one that's going to evolve to be very, very important in terms of utilization of these models. And in fact, we're trying to develop models where we can actually exploit the CRISPR-Cas system to look at collaborating oncogenes in the context of essentially a whole animal screen. And something that was really pioneered by a few people in the States to really get at this cooperating oncogene concept. So clearly the future remains quite bright for the development of these genetically engineered mouse models. I mean, the only thing I would say is that it does require a degree of patience and time to really examine these phenotypes in the depth that is required. So yeah, the Cree-Lox system is definitely another one, and I would say the CRISPR-Cas system would be a second major development that's really going to catapult this field into future discoveries.
One more question. There's a lot of trainees on this attending this workshop, and I'd like for you to give them a sense of perspective of the cost of the experiments back in the 1980s versus the cost of the transgenic or construction of a mouse model nowadays. I mean, it must have gone down dramatically.
Oh, it has. I mean, you know, in terms of the technology, back when I first started in 1986, there were literally less than 10 labs doing this. You know, and in fact, a lot of the history derives back to, you know, to developmental biology labs, of course. Martin Evans and Beatrice Mintz, who were doing a lot of embryo injections, and that's how transgenics got initially developed. I think the first was actually with Beatrice Mintz's lab, where they tried to put an SC40 into a developing embryo. So that was kind of the first prototypical attempt to actually develop a transgenic mouse. Subsequently, Rudolf Janisch, who was a postdoc in Beatrice Mintz's lab, further pioneered these efforts in embryonic stem cells. So there's been essentially coalescing of different technologies, which has enabled the field to be further advanced. I mean, development of ES technology is another significant advancement that's enabled us to knock out genes. And of course, with CRISPR-Cas, we can do that quite readily within a month time frame. To give you an idea of how fast things have progressed, we recently generated a knockout of a particular molecule we're interested in FEB with the transgenic facility. And Mitra will probably describe some of the stuff we've done in collaboration in literally 2 months, whereas back in my day it was taking 2 years. So the technology has increased in utility and economy. So things have moved quite a bit faster. Although, I'm doing the standard mouse genetics, it's still doing the standard mouse genetics. It takes time to actually get the appropriate combination of strains. But nonetheless, the technology that's enabled us to do these things is really dramatically advanced. And I would recommend anybody in the center to take advantage of the facility that we have. We have one that's probably, I would say, one of the best in North America.
Okay, thank you very much. Bill will be back at around 11:40 after Mitra's presentation if there are other questions. So we'll now move on to the second presentation in the workshop, which is going to be given by Dr. Yojiro Yamanaka of the Goodman Cancer Center. It's going to be on the CRISPR knockout models, which Bill has been talking about relative to recent developments. So